I'd like to thank the previous speakers who have made things a lot easier for me, because they explained phenomena which it took me a long time, first of all, to verify, and then the explanation given by physics. Now, the problem of our foundation, of course, is AIDS, but also other chronic diseases, which are just as important in terms of public health worldwide. And I'm referring in particular to chronic diseases. And for a long time, I wondered how germs, in the presence of highly active treatments, also in the presence of a highly efficient immune system, could persist in a body for years on end and cause terrible diseases. And one of the terrible diseases I studied is Lyme disease, which is a disease we can have an acute state, but usually moves to a chronic state where the polymorphic germ persists without being eliminated. So I studied new techniques and technologies and came across a phenomenon which I'm going to expose today. One experiment, but it encapsulates all the long years of work together with my colleagues, during which we tried to investigate the origins of the persistence. So I worked on the Lyme agent called Borrelia burgdorferi after Mr. Burgdorfer, and it is transmitted through bites, but also possibly through other forms of transmission. And therefore it's an insect bite which injects a certain number of bacteria into the body, about 30 species. But there may be other species which are involved in the disease. Now, to detect the persistence of the germ, what we did was use a highly sensitive technique, which is PCR, polymerase chain reaction, which can detect DNA triggers, and it's polymerase, which is thermoresistance. So it can be used for the fusion of the double helix. I also used a technology based on the production of electromagnetic waves. This was a most unexpected phenomenon for me back in 2005-2006. Now, this is the principle of PCRs. So you have a Taq polymerase on one of the DNA change with the trigger and oligonucleotides with replication of the new double helix. So the emission of electromagnetic waves brought about quite a lot of controversy and criticism, but these are very simple phenomena. Almost anybody is able to observe this. All you need is a measuring device, an amplifier, and a computer. That is enough to detect and then to record the waves. Now, what was surprising was that in filtrates of bacterial cultures, not only Borrelia but others as well, we detect the production of the waves at very low frequencies, and also in the plasma of people infected with the same agents. For instance, people who have contracted Lyme disease with the Lyme agent and detection can be achieved as quickly as with PCR. PCR doesn't always work, and this way you detect signals in most chronically affected patients. Now, everything is highly codified. You need dilutions of the filtered culture, and the DNA extracted from the cultures we saw could work in the place of the extract, and you could use frozen preparations, whereas with plasma you always need fresh plasma. That was one of the first observations which led us to think of structures which are labile in water. Heating makes it possible to eliminate the production of signals. Then there's filtration, which helps to pinpoint the structures producing the signals. Most of the structures pass through the 100 nanometer filters. These are the structures producing signals from bacteria and viruses, small viruses at least, induce structures which pass the 20 nanometer filters, so they're smaller. So by differentiating filtration from plasma, we're able to say whether it's a bacterium or a virus without being able to go any further, and I'll explain why. And for each dilution, there has to be a strong vortex shaking. And of course the question arose, what is the role of the shaking? Apparently it produces small nanobubbles which on the surface of the water might organize those structures which were mentioned by the previous speakers. So here you have a diagram which summarizes the experiments we run thousands of times. So we have the filtration. The dilution is 2 nanograms per milliliter, and then dilution. And if we're lucky to have structures which produce signals, we find them at quite high dilutions, 10.8 to 10 to minus 13, but the frequencies are extremely low. Or natural waves, Schumann waves, and the first harmonic peak is 7.83 Hertz. So it is a phenomenon of resonance. Here you have the responses to excitation with changes of amplitude and changes in frequencies. So here on the right, this is not an ECG, it's a different scale. These are milliseconds, and the spectrum is quite complex, and it is essentially an amplification of background noise. So you have the amplification of background noise, so you can measure an amplitude which is generally increased, and here in milliseconds at the top you have the background noise, and here you have 10 minus 3, 10 minus 4, and it's only bottom left that you have positive emission. And once again, some dilutions do emit signals and others don't. So we have a production of waves. So what happens? Obviously, we simplified these illustrations. They're not really true to reality, but it is a way for us to explain why at low dilutions you have a kind of network in liquid water which can be an agglomeration of coherence domains. And it's only after dilution that you have coherence domains which are free and which emit signals. Then how does the system self-maintain itself? Well, that was the surprise. It is through the emission of these waves, because they in turn induce matter structures. So this could be highly labile, with chemical bonds, H bonds in particular, but they can, in fact, endure for several hours and then slowly meet up again with the background noise. But this can be recorded, digitized on a computer, so we have a way in which we can maintain them in a stable state. Second surprise, an even bigger surprise in many ways, these structures maintain the information contained in the DNA. Benveniste worked on biological transmission. Here we are working on chemical structures, which are the DNA sequences which can easily be verified. So based on these water structures, we can track back to the DNA structures that existed initially. So how can you tell the structures exist in water? Well, filtration first and foremost. So we have evidence of their size, and also more recent work together with Dr. Tsenkova from the University of Kobe. Infrared was used with nonlinear methods to see the subtle spectral changes related to DNA dilution. And the surprise there was that the change in structure observed is observed only in dilutions which produce electromagnetic signals. So clearly there are changes in the structure of water, probably as Dr. Vitiello and Dr. Henri were saying, there's a constitution of coherence domains which are relatively stable and self-maintain in an incoherent, non-coherent medium. So these structures we've called nanions. And I'm going to show now that these structures carry DNA information. So 3 stages here. 2 have been published, the 3rd is new. First experiment. We put close together a tube of DNA with a dilution which produces electromagnetic signals. That's tube 1, and it is put in a container which doesn't let through external electromagnetic signals, next to a second tube which contains water. The 2 tubes are then exposed to a generator, 7 Hertz, so we induce a magnetic field in the solenoid within the shield, and after 18 hours we take a look at what is happening in the tube of water, and we dilute and we see that there is an emission of electromagnetic signals. So the information from tube 1 is transferred to tube 2, and there's no contact, so it is through waves that the information was conveyed. So if we then perform PCR on the tube of water at the bottom, at the top you have tube number 1, which is the control with DNA dilutions, but very quickly PCR switches off at dilution level 4, but the recipient tube, tube number 2, shows a dilution which goes all the way down to -8. So information has been transferred, and in fact it is more persistent in the water tube than in the control tube. So what we have here is a piece of HIV DNA. It's the LTR fragment. 104 base pairs here. On the left you have the molecular weight markers in an electrophoresis gel. So through sequencing, with the 104 base pairs, we have the composition of LTR. So with this experiment, obviously we were working in a laboratory. People say, well, yes, you contaminated through PCR, but we took every precaution. We eliminated all risk of contamination, but we felt that we also had to work with a remote laboratory, a laboratory which had never worked on the DNA and the virus we were working on. So we recorded the signals from a piece of DNA. It can be HIV, but we also work on Borrelia and the Lyme agent, and this is then digitized by computer. Then it is sent via the internet. The file is then received by the computer in the remote laboratory, then transformed into an analogical signal and amplified and transformed into electric current in a solenoid here in blue, which in turn will emit a magnetic field. A tube of water is set inside the solenoid, and after a short while, about an hour after the signal is received, a fragment of the water is taken, and with PCR the DNA is retrieved. And this was not done by us, but by a student in the laboratory who was an intern, didn't have much experience, but learned a lot very fast by working in the laboratory, and got well acquainted with the technology. So it's relatively easy to reproduce. There's also the laboratory in Belvento, which did the same thing for HIV-1 LTR. So this is the result of the electrophoresis in Germany, in the German laboratory, and you see it is very clear. And here you have all the components of the system. They're all present. The PCR, the nucleotide. Now this is really amazing, because how can a polymerase read nanostructures? Now we haven't fully solved this one yet, but the facts are there. It is possible the polymerase can read something which resembles DNA structure, but isn't DNA. It's a structure of water which has retained information. So these are well-controlled stages. They are reproducible, but haven't been totally elucidated. And I'm counting on our colleagues who are physicians and physicists and mathematicians to help elucidate this. And I shall now come to the third stage of this experiment, which is the most intriguing, it must be said. So we started with the amplicon, that is that pass of ribosomal DNA amplified by PCR, so millions of identical molecules. Regarding DNA, it is a coherent structure itself, and we stored it in digital form on a computer, and then we moved on to a second stage. The question was, are there other polymerases other than Taq polymerase which can also read these structures. So we wondered whether cultivated human cells had this ability. And the answer is yes. But these are tumor cells so far. So what we did was, based on the recorded file, made analog, amplified, we put electric current into a solenoid around the culture cells. These are cells suspended, so they are at the bottom of the flask, and we induced a modulated magnetic field, both in the medium and the cells. And after several days' exposure, what we have is a biological effect, and we reconstituted the DNA in the cells. That is the inhibition of growth in the cells. Here you have the cell count in green, the live cells, and in red, the dead cells. At the top, you have T0, and at the bottom you have the situation after a 4-day period. The number of living cells has decreased considerably. The number of dead cells has increased considerably, with a smaller size. The size has diminished in the cells killed by exposure. So alongside the phenomenon, we extract the DNA from these cells at different times on days 4, 5, 6, and 7, and through PCR we have the ribosomal DNA of Borrelia. The cells containing the DNA, and if you take control here, for instance, the signals are registered by the culture medium itself, you have nothing. And these cells, once again, these are lymphoma cells, U937, but we experimented with other types of cells as well. Breast cancer cells and leukemia cells. This is a table which I think summarizes what I was saying a couple of weeks ago, because there are findings every week, and I must say they're always the same. So on the left you have the files, ribosomal DNA of Borrelia, then ribosomal DNA of a cofactor of HIV, but I shan't go into that. And then the same sequence with the bacterial cofactor of HIV, but of human origin, and it's the genomic sequence does not give any signal. And then you have a Suturella ribosomal DNA associated with autism. If I have time, I'll discuss that too. But here again, we do not have any effect in the tumor cells, and no DNA. Now, the reasons are unknown so far, but what I am saying is that there is a specificity of signal. A lot of work has been done exposing cancer cells to a variety of frequencies. But here we have something which is modulated and which carries information with a biological effect. Now, you don't see it here in this table, but we also compared normal cells, diploid C5 cells, and T lymphocytes activated in culture. And those cells do not die and do not carry the DNA information. Now, I'm not saying that we can cure cancer, but it is definitely a direction we can continue looking into, because we have pluripotent cells. Professor Carlo Ventura also can look into the question, will these DNA cells die or not? What we can say is that normal mice exposed to these frequencies, very low energy, are not affected. So that is what I wanted to say. And to conclude, maybe a few words about application which is far more advanced, that is the measure of electromagnetic signals in autism. The assumption here, which of course does not exclude others, the assumption is that in autism there are organic infectious factors at play, and that those factors come from the intestinal flora. And we think that the gut floral, due to a number of factors, environmental factors, become open to pathogen bacteria, and are then found in the blood, the plasma of these children. And traces have been found of a group of bacteria identified by Ian Lipkin's group with a PCR technique. In the small intestine, there were considerable increases for this group of bacteria. The first species is Suturella, but there are others which are also known. And this compared to other children suffering from the same intestinal disorders, but not autistic. Now, this is an example of the production of signals in the plasma of an autistic child treated by antibiotics, azithromycin and cefuroxime, with clinical improvements. I'm not talking of cure here, but I'm talking about improvement. And if you look at the PCR, you also see this bond of Suturella DNA present in all children, which sometimes drops. M0M question mark here, after several month treatment. The cohort of children was 150 autistic children against 33 healthy children, and in the autistic children 85% have signals, and 82% are positive to the germ. We also tested for Borrelia. There are positives, but the difference is not significant. So what you can say is, based on these results, is that there are immediate clinical applications here, and clinicians are trying to devise the best possible treatments with antibiotics, but also other combinations which could diminish the signals. The production of signals, in a way, are the marker of the therapeutic effect before even any clinical improvement is observed. And to conclude, I'd say that this model could also apply to other neurodegenerative diseases such as Alzheimer's or Parkinson's. It is a slow, complex development. There are environmental factors, genetic factors, and they bring about modifications, biological modifications, and can sometimes create oxidative stress. And immunosuppression, at least partially, and therefore it passes into the blood, and then there are somatic mutations affecting DNA, and then possibly the production, and this I didn't say earlier, the oxidation of DNA. It's a recent observation we made, but which is also based on Jackie Barton's group in California's work with the transfer of electrons over large distances, the production of the movement of free electrons could also be involved. So as the disease progresses, it might in fact increase the effect of the bacteria, and this is how pathogenic information can persist and also escape immune defense and treatment. These nanostructures can be retranscribed in DNA and other cells because some cells do have polymerases able to read the structures. This is, in fact, a description of natural phenomena. It's not just tinkering in laboratories. So what I wanted to say is that this work, which of course begs a number of further questions: how to regulate the production of signals through DNA oxidation, how to apply a treatment based on the waves emitted by the structures, all that still lies in the future. But what I do want to say here is that we're doing something which is solid. It's not something which is confined to one laboratory. Many people are working with us, also physicists. Professor Carlo Venturo is also going to describe some amazing things related to cell differentiation and the music of cells. And I think maybe I should let him talk about it himself. Thank you.